Hello everyone, I'm Li Chen from University of Washington, presenting for the paper of automated intracranial artery labeling using a graph neural network and hierarchical refinement. Intracranial arteries are a large complex network of more than 100 artery segments. Although there's a large individual variations of intracranial arteries, the anatomical names of arteries can still be identified by human. For example, on the right figure shows different labeling methods for intracranial arteries. Many segmentation or tracing algorithms have been developed to quantify artery features from MR angiography, but regional vascular feature quantification requires labeling each artery accurately. Among all the arteries, the 9 so-called Willis arteries, forming a ring in the middle of the intracranial arteries, attract most attention from the clinical perspective. With correct labeling of so-called Willis, we can assess flow conditions in anterior or posterior circulation and evaluate left-right flow balance. So-called Willis is also important because it is the region likely to have vascular disease, and correct identification of its anatomical variation can provide useful geometric risk factor. Considering the manual labeling is not feasible, especially for medical research on large population studies, an automated labeling method is critically needed. Previous methods in solving intracranial artery labeling can be summarized into two approaches. First is to generate an atlas from training sets that match with unlabeled graph. The second uses statistical methods to estimate the probability of each type of bifurcation point in the intracranial artery network, then to label the arteries based on bifurcations. Currently, there is only the UNC dataset, which has 50 healthy subjects publicly available for intracranial artery labeling. Many existing methods are tested only on this small dataset with limited anatomical variations. In addition, the global constraints for the relation between arteries are not usually considered. To solve the intracranial artery labeling problem, we propose a new approach by using graph neural network followed by the hierarchical refinement. Intracranial artery graphs are constructed from artery traces, then sent to a graph neural network to predict the node and edge categories. Then, a hierarchical refinement framework is used to correct the prediction mistakes from the graph neural network predictions using the anatomical prior knowledge while maintaining flexibility to intracranial artery variations in a hierarchical order of 3 levels. We use MICCAI, a semi-automated artery tracing tool, to acquire artery traces from top MRA. Artery traces are a natural graph, but for simplicity, only key nodes with degree other than 2 are used in the graph neural network. Our aim is to predict the node and edge categories which are already labeled by human as ground truths following the anatomical labeling definitions in MICCAI. The exception is we ignore the M2, M3 node labels due to the largely debatable criteria. We collected the largest dataset for ICA labeling from 5 of our research projects or collaborating hospitals. Compared with the existing UNC dataset, our dataset includes 5 sources having different imaging protocols, coverage, vendors, care state, image sources, which include far more intracranial artery variations, and thus is more challenging for automated labeling. The graph neural network takes a graph with node and edge features as input and returns the graph as output with additional features for node and edge categories. The input features of edges and nodes in the graph are encoded to an embedding in the encoder layer. Then, the core layer passes messages for 10 rounds by concatenating the encoder's output with the previous output of the core layer. The embedding is restored to edge and node features in the decoder layer with additional label feature. In each graph block, the edge attributes are updated through the per-edge update function phi-e. The features for edge connected to the same node are aggregated through function rule to update node features through the per-node update function phi-v. Input features used in the graph neural network include normalized XYZ coordinates, degree, radius, and directional embedding of the node. Due to the uncertain degree of node in the bifurcation, we represent the direction of branches in a 26-dimensional embedding. Each branch is matched with the closest 26 major directions with 45 degrees apart in a sphere, then 26 directions are encoded into 26 binaries based on whether a branch is matching. Edge features include edge direction, distance between nodes at two ends, mean radius at two nodes. Output features for graph neural network are one hot encoding for 23 edge types and 20 node types, shown in the figure on the right. Please refer to the Acknowledgements paper for detailed definitions. End-to-end graph neural network predictions are not the optimal solution for labeling, as nodes and edges predictions may not be compatible. Considering manual labeling on intracranial arteries is sequential, for example, labeling large major branches before small optional branches. Automated labeling should also be hierarchical with combined information for nodes and edges. Here, we propose to post-process the graph neural network prediction in a three-layered hierarchical framework, starting from the most confident prediction. From graph neural network predictions, each node and edge are predicted as a certain type with largest probability. In the first layer, we identify most confident nodes as the nodes with fitting predictions. For example, the blue node is predicted as ICA, MCA, ICA left, with three of its branches predicted as ICA left, A1 left, and M1 left, which fits the anatomical definitions of this node. So the blue node is considered confident. Similarly, for the red node if the node is predicted as ICA root left. In the second layer, the intracranial artery tree is subdivided into three subtrees, each with major nodes and branch nodes. For example, for the anterior left cerebral tree, ICA, MCA, ACA is the major node and the branch nodes are its three neighbors. If the major node is not confident node, we first find the best node to be the major node, then predict the rest of the branch nodes which are not confident by minimizing the combined probability for nodes and edges. In the third layer, auxiliary branches such as second-order MCA are labeled from the already labeled nodes. Some branches likely to be missing due to variations can be handled with flexibility in the framework. Here we show some examples with rare intracranial artery variations but predicted correctly by our methods. First is a subject with Parkinson's disease. Occlusions cause both right and left internal carotid arteries to be partially visible. In addition, PCOMs are missing. Second is a hypertensive subject with rare A1 left artery missing. On the left, some lenticular stride arteries are visible in our dataset. That's an additional challenge for labeling, but our method predicted it correctly as a non-type. On the right, it shows even if we add more artificial lenticular stride arteries, our method is still robust to this added noise. We use the following metrics to evaluate the performance of our method quantitatively, including accuracy of predicted node labels. To divide the performance of labeling in each bifurcation, detachment accuracy, precision, recall for each bifurcation type is also evaluated. Evaluated on our dataset, our method is more accurate and takes less time compared with existing methods. As an ablation study, the directional embedding and hierarchical refinements are crucial for the best performance in our model. We also evaluated our method on the relatively easy public UNC dataset and cite the performance from existing works. Even without training on the UNC dataset, our method is able to achieve 92% accuracy. If used as threefold cross-validation, our method can outperform previous methods, even if they are using more training data with leave one out cross-validation. In this work, we have constructed a large and diverse public dataset for automated labeling. With 729 scans, including many rare and challenging intracranial artery variations, this can be a new benchmark for automated labeling. We have explored the ability of graph neural network to learn from the graph representation and label the arteries. And we also found the performance can be further improved with hierarchical refinement to incorporate prior knowledge about intracranial artery structures. Our method is fully automated. Within 0.1 seconds, 20 node and 22 edge types can be predicted, so that comprehensive features for detailed analysis of cerebral flow can be acquired. In addition, the framework is also applicable to any graph structure where sequential labeling helps. Our datasets and codes are publicly available in this link. We acknowledge the support from National Institute of Health and the following researchers. We acknowledge NVIDIA for providing the GPU. Thanks for your attention.